Okay, so excitation contraction coupling is how we use calcium to regulate this process. It's a sequence of events where an action potential arrives at the muscle membrane, the sarcolemma, and that eventually results in contraction. This is going to depend on the motor neuron, so the innervation coming from the motor neuron, that whole series of events at the neuromuscular junction, along with the release of calcium from the sarcoplasmic reticulum. We talked about this, the unique name for some of the organelles in muscle. And so the endoplasmic reticulum is the term that's used in other cells for that organelle that stores calcium. And here for muscle, we call it the sarcoplasmic reticulum, where it's doing the exact same job. It's pretty much storing calcium for release at a specific time. So this slide is pretty much a review. This is talking about what's happening at the neuromuscular junction. We've got a motor neuron that's innervating a muscle cell or muscle fiber. We spoke about the fact that each muscle fiber can only be innervated by one motor neuron. And then one motor neuron can innervate many muscle fibers. And that was what comprised a motor unit. We spoke about the events that happen at a synapse. So it's similar events that are happening here as far as the action potential getting to the axon terminal, the exocytosis after the rush of calcium into those voltage gated calcium channels, triggering that exocytosis, the acetylcholine being released, binding to our nicotinic receptors that are at the motor end plate. And we spoke about some of the features of the motor end plate. We've got the nicotinic receptors there. It's highly folded, and the excitation of this membrane is called an end plate potential. So an end plate potential is basically the action potential, the result of the action potential on skeletal muscle. We said that it's always strong enough to bring about muscle contraction. We also said that it's always excitatory. There's no such thing as an inhibitory response on the end plate, on the motor end plate. Always excitatory, always strong enough to bring about or generate muscle contraction. And that is going to basically excite the muscle. So this is one of the only spots in the nervous system where we see that chemical energy being transferred, not to electrical energy the way that we see it on other neurons, but to mechanical energy. Energy that can be used to actually do physical movement. So we go from electrical energy on the neuron to chemical energy at the synapse to mechanical energy at the motor end plate, okay? All right, let's speak about how calcium is involved in all of this. Calcium is involved as it pertains to the two regulatory proteins that are involved with actin. So we introduced these regulatory proteins, they're troponin and tropomyosin. Troponin being that complex of three proteins that are binding calcium. One binds calcium, one binds tropomyosin, and one is binding onto actin. And then tropomyosin is the thread-like regulatory protein that is covering up those myosin binding sites on the actin filament. So if there's no calcium in the sarcoplasm, so in the cytoplasm of the cell, in muscle we call it the sarcoplasm, if there's no calcium present, troponin is going to keep tropomyosin, covering up those myosin binding sites on actin. And as long as those myosin binding sites are covered up, we cannot make interaction between actin and myosin. In other words, we cannot form cross bridges. We cannot, initiate that cross bridge cycling. In this state, muscle is relaxed. It's in its relaxed state because there's no calcium present. When calcium is present, and we'll talk about how that actually happens, but calcium becomes present as a result of the end plate potential. So it's kind of the same sequence of events here. The sarcoplasm is flooded with calcium. Now in the presence of calcium, it can bind to troponin. That's going to cause troponin to drag or slide tropomyosin off of those binding sites. It's going to expose those myosin binding sites on actin such that myosin can now reach up and make interaction with those binding sites on actin. This is where we can now initiate the cross-bridging, right, from step one of the cross-bridge cycling. And this is where we can begin to shorten or contract the sarcomere, which brings about the muscle contraction overall. So that's how calcium regulates this whole process, okay? Let's look at the steps of excitation, contraction, coupling. So there are a couple of steps here, and they kind of lend or begin with the neuromuscular junction event. So that action potential, arriving at the sarcolemma, generating that end plate potential. And from the end plate potential, we can pick up the conversation, speak about how that action potential, and you can insert the word end plate potential here, because the end plate potential is really what's happening specific to skeletal muscle. So those are pretty synonymous. So the end plate potential, the action potential, can now travel deep throughout the muscle from the sarcolemma, which is really the membrane of the muscle, via the T-tubules. So if you remember us speaking about those transverse tubules, those deep invaginations that run throughout the muscle, the function of those structures is to help the action potential get deep within the muscle to propagate that electrical energy in order to generate the excitation of the entire muscle very quickly. It's going to call on two receptors, DHP receptors, dihydropyridine, we'll talk about those in a bit, that are associated with the T-tubules, along with lyanodine receptors, which are also coming in contact with those DHP receptors. It's also going to call on that triad. We spoke about that structure. It's a collection of one T-tubule and two lateral sacs on either side, or the terminal cisternae, we also call those structures, that created this triad of structures. Those players are involved in the propagating of the action potential and how calcium becomes the result of the action potential as it travels throughout the muscle. So when that whole, communication of signals happens, the calcium is then released from the sarcoplasmic reticulum where it's being stored into the cytosol or the sarcoplasm, right, same terms. And then that calcium can then bind to troponin, bring about that shift of tropomyosin, and then we can start cross-bridge cycling. All righty. Okay, so let's walk through those events here. I'll pause and see what questions you've got first. So I'll take a brief pause here, see if you have any questions about cross-bridge cycling, about the players in the excitation, contraction, coupling, and then we'll kind of go over it again here a second time. All right. So we're starting off from the neuromuscular junction, a series of events that we've spoken a bit about back in the somatic nervous system. We've got ACH, which is acetylcholine, released from our axon terminal of our motor neuron. Remember, this is a motor neuron coming from the ventral horn. Remember all of that, all of those steps that are occurring before this synaptic event. It releases its neurotransmitter, crosses that junction, binds to this nicotinic receptor. Although they're not specifying that this is a nicotinic receptor, we want to be mindful of that. This can only be a nicotinic receptor. Binds to that receptor, generates here an end plate potential. Always going to be excitatory, always going to have enough amplitude to excite this membrane, okay? Then that leads to the propagation of this action potential throughout the entire muscle. So because it's starting from the membrane, it's really important to get this electrical energy deep throughout the muscle really quickly. And that is the purpose of our T-tubules, those transverse tubules that are really these invaginations or tunnels that run deep throughout the length, the width of the muscle rather. After we have the propagation of that energy, that action potential throughout the T-tubules, throughout the muscle, the T-tubules are actually adjacent to the channels that are on the sarcoplasmic reticulum. And so when the action potential gets to these structures, it's going to facilitate the release of calcium from the sarcoplasmic reticulum. So the sarcoplasmic reticulum is shown here in blue. That's where calcium is being stored. And we need to get that calcium from its storage area here out into the sarcoplasm of the cell itself so that it can interact with troponin to regulate the process. So now we've got the calcium out into the sarcoplasm, binds onto troponin. When it binds to troponin, troponin is then going to drag tropomyosin off of the binding sites. And it's really important to remember here that troponin is a series of 3 different proteins. So when we bind one spot with calcium, we have a conformational shift of the entire structure such that the other site that has myosin bound to it can shift, and that's what drags the thread-like protein tropomyosin off of those binding sites which were otherwise covered up. So now we've exposed those binding sites. The myosin head can now reach up, grab on, or make interaction with that site, bring about that cross-bridge cycling with the power stroke and the other processes involving ATP. And then we have to end this process off in a very similar way that we needed to clear the neuromuscular junction in order to cease this event. We likewise have to kind of drive all that calcium back into the sarcoplasmic reticulum in order to cease muscle contraction. Okay, we've got to dump or clear the excess calcium in order to cease muscle contraction and to allow the troponin to reposition tropomyosin to cover up those binding sites once again in order to break the interaction between actin and myosin and bring the sarcomere back to its relaxed state. Okay, so here we can see once we clear that calcium from the sarcoplasm, drive it back into the sarcoplasmic reticulum, we then allow, because this troponin doesn't have calcium on it, it's then going to bring back its original position where it drags that thread-like protein to cover up those sites, and that's how we can relax muscle. Okay, and so here we can look just at the two states or the two positions of actin and the regulatory proteins that are associated here. So here's the actin. This is what's actually and I use the word contractile here loosely, but it is considered a contractile protein because it is what's moving to bring about the shortening of the sarcomere, but we want to understand that it's not actually contracting or shortening in and of itself, but it is considered a contractile protein. Whereas tropomyosin and troponin are not contractile proteins, they're regulatory proteins because they're just regulating or turning this process on or off. Okay, really important distinction. So we can compare the relaxed state of muscle where troponin is not bound by calcium because there's no calcium in the sarcoplasm. And so it's keeping the tropomyosin attached to these binding sites and preventing myosin from making an interaction. And then we can compare that to the contracted state of the sarcomere or of the muscle itself, where calcium is now present. It's washed out of the sarcoplasmic reticulum. It's now in the sarcoplasm. It's going to bind onto its spot on troponin. It's going to allow the entire complex to drag the tropomyosin protein off of the binding sites. And now we can have the myosin sites exposed. Myosin can make an interaction and begin the cross-bridge cycling. So this is how calcium is essential, crucial to turning this process on, but also very, very important, it has to leave the sarcoplasm in order to turn this process off.